Hi everyone, welcome to today's lecture where we will understand about one of the essential techniques in microbiology, capsule staining. Now you might already know that bacteria can have various external structures, but one of the most important when it comes to infection and disease is the capsule. This outermost layer plays a crucial role in helping bacteria survive and evade our immune system. But here is the interesting part. Capsule don't take off stains like the rest of the cell. So how do we see them under a microscope? That's where the special staining method comes in. By the end of this lecture, you will not only understand how capsule staining works, but also why it is so vital in diagnosing infections caused by bacteria like Klebsiella pneumoniae or Streptococcus pneumoniae. So let's get started. Let's begin by understanding what a bacterial capsule actually is. A capsule is a thick, well organized layer that surrounds some bacterial cells. It's usually made of polysaccharides, that's sugar molecules, but in some bacteria like Bacillus anthracis, it's made of polypeptides, specifically D-glutamic acid. What does the capsule do? It acts as a protective shield. It helps the bacteria escape phagocytosis, which means our immune cells can't easily engulf and destroy them. It also prevents desiccation or drying out and helps the bacteria stick to the surfaces and tissues. These features make the encapsulated bacteria more virulent, or in simple terms, better at causing the disease. Now let's understand the principle behind capsule staining. Unlike the bacterial cell wall, the capsule is non-ionic, meaning it does not attract or bind to most commonly used stains. So when we try to stain a bacteria with the traditional dyes, the capsule remains invisible. To solve this, we use a dual staining approach. First, we stain the background using an acidic dye like the India red, India ink, or Congo red. These dyes don't penetrate the capsule or the cell. Next, we apply a basic dye like crystal violet or safranin, which stains the bacterial cell. As a result, under the microscope, we see a dark background, a stained bacterial cell, and a clear, unstained halo around it, which is the capsule. Before performing capsule staining, it's important to gather all the required materials. You will need a bacterial culture that produces a capsule. Klebsiella pneumoniae is the common choice. Then you will need clean glass slides, an inoculating loop, and two types of stains, an acidic stain like India ink and a basic stain like crystal violet or safranin. You will also need distilled water for rinsing, a staining tray, and a good quality microscope with oil immersion capability. Here is one important note. We do not heat fix the slide during this staining process because heat can damage or distort the capsule. This is crucial point for students to remember. Now let's walk through the step-by-step procedure for capsule staining using the India ink method. Step 1. Place a drop of India ink on one end of a clean slide. Step 2. Mix in a small amount of bacterial culture using a sterile loop. And in the third step, use another slide at a 45-degree angle to spread the mixture like a blood smear. Allow it to air dry. Do not heat fix. Next, stain the smear with crystal violet for about 1 minute. This stains the bacterial cell. Then rinse the slide gently with distilled water and air dry again. Now it's time to examine your slide under the oil immersion lens of the microscope. If done correctly, you will see cells surrounded by bright color halo, which is the capsule. So what should we expect to see under a microscope after capsule staining? You will notice a dark colored background due to the India ink or Congo red. Within that, the bacterial cell itself will appear purple or pink depending on whether you use crystal violet or the saffron. But the most important thing, you will see a clear colorless halos around each cell, that is the capsule. This visualization confirms the presence of a capsule and helps differentiate encapsulated bacteria from those without capsule. Let's now look at some important capsulated bacteria you should remember, especially for exams or any kind of clinical relevance. Klebsiella pneumoniae causes pneumonia and is have a very thick capsule. Streptococcus pneumoniae, a major causes of bacterial meningitis and respiratory infection. Bacillus anthracis causes anthrax, has a unique polypeptide capsule. Neisseria meningitidis, another key meningitis causing organism. Haemophilus influenzae, often found in childhood respiratory infection. In many of these organisms, the capsule is a key virulent factor, meaning it plays a direct role in causing the disease. Now, you might be wondering, why is this technique important? Here are some real world application of capsule staining. It's used in clinical microbiology to identify pathogenic bacteria in patient samples. Helps understand bacterial virulence mechanism in research. It's also essential for developing vaccines against encapsulated bacteria, as the capsule polysaccharides are strong antigens. Also used to differentiate between strains of the same species based on presence or absence of a capsule. So even though it seems like a sample lab method, it has critical diagnostic and research value. To wrap up, let's summarize what we have learned today. Capsule staining is a method used to visualize bacterial capsules that are otherwise invisible under the standard staining. The capsule is a protective non-ionic structure that plays a key role in bacterial survival and virulence. Using a combination of acidic stains for the background and basic stains for the bacterial cell, we create contrast where the capsule appears as a clear halo. Understanding and applying these techniques is essential for identifying pathogens in a clinical setting and for mastering microbiology lab skills. Thanks for staying with me through this session. If you found this lecture helpful, don't forget to like, share, and subscribe for more microbiology tutorials. See you in the next lecture. Thank you.